In today's lecture, we're going to make another major transition in our course on the science of life, as we shift our focus to the third and final conceptual theme around which the course is organized, the theme of energy and resources. Now, before we begin, let me take this opportunity to remind you of the overall organization of the course to help us see where we've gotten to so far as a way of introducing where we're going next. There are many ways that a first university course in biology might be organized, depending on the goals of the course. Our purpose in the science of life has been to emphasize the unifying principles of biology, principles that are useful for gaining an appreciation of the subject as a whole, as well as for providing a framework we can use to help us interpret and understand the many things we encounter in our daily lives that have to do with biology in one way or another. To this end, you'll recall, our course is organized into three major sections, with each section focused on a unifying conceptual theme, a conceptual theme that's central to understanding how living things work. You'll also recall that within each of these three conceptual themes, our approach has been to explore that theme across levels of organization in the hierarchy of living systems, considering each first from the fundamental level of molecules and cells, and then building our understanding up through progressively higher levels of organization, at least as much as we can do that conveniently. Let's go back and review briefly where we began with the first third of our course, which focused on the broad theme of information and evolution. Now here, we started with the question of how living things could store and transmit information about their own structure so that they could make more of themselves, an ability that's absolutely fundamental for life to exist. This information, we learned, is the information about the structure of proteins. These proteins, of course, being the molecular workhorses of the cell. And we learned that the nucleic acid DNA is the molecule that's responsible for storing this information. DNA does this, you'll remember, by having a unique sequence of nucleotide bases that correspond through the triplet code to a unique sequence of amino acids that make up a particular protein. We spent a considerable amount of time looking at the structure and function of DNA, especially at how it's replicated, how it's transcribed into RNA, and how this RNA is translated into a protein. We also saw how replicated DNA is divided into two copies in an orderly fashion into the two daughter cells that are produced when a particular cell divides. This was the process of mitosis, which ensures that both of the daughter cells get a complete complement of this genetic information. Now, we were particularly interested in a different kind of cell division, one that occurs when cells undergo sexual reproduction. You'll remember this was the process of meiosis. In meiosis, we saw how the movements of homologous chromosomes, those are the two copies of chromosomes that are inherited from parents, one from the mother and one from the father, how the movement of these homologous chromosomes could account for the ways that traits are passed from parents to offspring. This observation led us to look more closely at these patterns or laws, which we referred to as Mendelian genetics, in honor of the monk who inferred them from his studies of pea plants over 150 years ago. Our discussion of Mendelian genetics, combined with our earlier discussion of molecular genetics, led us to an appreciation of how genetic variation, resulting from mutation and genetic recombination, is constantly introduced into the genetic makeup of organisms, such that offspring will never exactly be the same as their parents. And we saw that the most profound consequence of this variation is that it provides the substrate for evolution by natural selection, an idea that was first proposed by Charles Darwin in the middle of the 19th century, and that's become since the single most important organizing idea in biology. We then spent a considerable amount of time in the first third of the course talking about evolutionary mechanisms, looking at how populations change over time, not only through natural selection, but also as a result of random genetic drift, and other factors that affect the relative proportions of alleles in what we call the gene pool of a population. Finally, we concluded our exploration of information and evolution in biology by considering how evolutionary mechanisms might lead to speciation, accounting for the diversification of organisms that has occurred over the vast extents of evolutionary time. And then we asked how we could organize in a systematic fashion and make sense of this diversity. Now, a point that I concluded that section of the course with, and I'd like to emphasize again, is that in a very real sense, the genetic information which is found in the innumerable different kinds of organisms that exist today or that ever existed throughout time is really just the result of the modification of the original genetic information that was found in the first cell that we might want to have called living very early in the primordial Earth. In the middle third of our course, which we've just completed, we turned our attention to a different organizing theme, the dual theme, really, of development and homeostasis. Actually, another way to state the unifying theme around the middle third of the course, around which the middle third of the course was organized, would have been in a question: what does it take to be a multicellular organism? In the broadest brushstrokes, we saw that it takes two things to be multicellular. First, it is necessary for a single cell with a single set of genes to be able to develop into a diverse set of cells, each having different characteristics, different functions, and different positions in a complex organism. This requirement led us to consider the problem of development. Second, multicellular organisms have to have a way for their various parts to communicate in order to coordinate the activities of these diverse parts, having diverse functions, and in so doing to maintain a favorable and stable internal environment. This requirement led us to consider the issue of homeostasis and its many manifestations. To explore the concepts of development and homeostasis, we began the middle third of the course on the molecular level of organization, considering first the problem of how different kinds of cells in the same organism can express different proteins at different times and in different places. Of course, the solution to this problem, you'll recall, is that not all the genes in a particular cell's genome are expressed all the time. And to understand this solution, we looked in some detail at the mechanisms which regulate gene expression. This discussion actually revealed a very important additional critical role of proteins, which is to serve as what we call transcription factors, essentially acting like molecular keys that lock and unlock the expression of particular genes. After we understood the basics of gene expression and how the proteins that are produced by the cell get processed, packaged, and sent to their proper destinations, we next turn to the question of how cells talk to each other in such a way as to control and coordinate each other's activities. That is, we turn to the problem of cell signaling. Here, we learn that the binding of signal molecules outside the cell to protein receptors that span the cell membrane can initiate a cascade of molecular events on the inside of the cell that activate or deactivate proteins, for example, by having those proteins become what we called phosphorylated. And this cascade of protein activation then in turn affects the way that the cell functions by turning on and off functional proteins that do something for the cell, or perhaps by affecting transcription factors which then affect the genes that the cell expresses. Armed with this background on gene expression and cell signaling, we then examined in some detail the question of how a single cell can develop into a complex organism. We saw here how really only a few basic processes, cell division, cell differentiation, and a bunch of so-called morphogenetic processes, including cell migration, the movement of cells, changes in cell shape, and programmed cell death, these morphogenetic processes all are responsible for the unfolding of a complex organism from a single cell. We also saw in this part of the course how different factors regulate these processes at different times in development. Specifically, we saw how proteins and messenger RNAs that had been packed into the fertilized zygote by the mother, we called these cytoplasmic determinants or maternal determinants. We saw how these cytoplasmic determinants largely control the very earliest phases of development, with the developing individual's own genes only playing a substantial role at some time later in development, with gastrulation in many organisms usually being the turning point where the switch in control occurs. We also saw, finally, how signals from other cells and tissues play an essential role in determining a particular cell's fate in a developing organism, with diffusible positional signals especially being important in establishing the complex three-dimensional morphology of an organism. Now, after looking at how multicellular organisms were built, we then turned our attention to how these multicellular organisms maintain themselves. That is, we turned to the concept of homeostasis. Homeostasis, you'll recall, requires the coordinated responses of effectors that counteract perturbations of physiological systems through negative feedback, and in so doing, return physiological systems to some optimal set point. Now, to be able to do this, we realized organisms have evolved sophisticated systems for transmitting physiological information. And our exploration of these systems led us to consider in turn how hormones work, how neurons work, and how nervous systems in general work in animals. Our use of homeostasis, however, in this part of the course was really quite broad, including not only the ability to establish and maintain a stable internal environment, as was originally envisioned by Claude Bernard in the 19th century. Remember, Bernard was the person who defined homeostasis first. But we also included the ability of an organism to maintain a favorable external environment as well, and to defend the inside of the organism from challenges coming from the outside. In this broader context, we considered how animals use sensory systems to transduce physical information obtained from the outside of the world into physiological information in the form of action potentials fired by neurons in the nervous system. And we also considered the molecular mechanisms that underlie the ability of animals, I should say, to move. That is, we looked at how animals were able to change where they were in the external world, how their muscles worked, which is potentially the most important adaptation that animals have for improving their situation. Now, this broad interpretation we took to homeostasis also led us to consider the defense mechanisms that animals have evolved to protect their internal environments from injury or attack by disease or parasites. That is, we turned our attention to how the immune system worked. Here's where we saw what was perhaps the most impressive example of different cell types and molecules acting in a highly coordinated fashion to achieve a common outcome. That is, the outcome of identifying and eliminating agents that might compromise the integrity and well-being of a complex multicellular organism. Finally, we ended the middle third of the course in the last lecture by talking about behavior, referring to behavior as a kind of highly elaborated adaptation for achieving an optimal environment. Now, before we go on and talk about where we're going next, let me point out something you might have realized already, which is that our discussion of development and homeostasis that we've just completed didn't really follow as exact a progression through levels of biological organization as we were able to achieve in our first third of the course about information and evolution. We kept jumping back down to the level of molecules and cellular mechanisms repeatedly throughout this part of the course, and we never really got to consider the issue of development and homeostasis at levels of organization higher than that of individual organisms. In other words, we didn't look at what it meant to think about development or homeostasis in terms of populations of organisms or communities of different kinds of species and so forth. Now, the main reason for this is, as I said earlier, because the middle third of the course really was about being multicellular. And so the organism itself appropriately served as the high point of our discussion. One might wonder, however, how the theme of development and homeostasis could be illustrated in the context of populations or communities, or even in the context of ecosystems. Actually, as we'll see, some of these issues will appear in the final third of the course. So let's move on and ask where we're going to go next. In the third and final section of this course, we're going to turn our attention to the theme of energy and resources. Now, to introduce this theme and its importance, let me go back to a couple of specific examples we've discussed earlier. Recall first the role of a molecule called transfer RNA in protein synthesis. This is a process that we discussed in the first third of the course in the context of what we referred to as the central dogma of molecular biology. Remember that transfer RNAs, or tRNAs for short, are the molecules that can talk both the biochemical language of nucleic acids and the biochemical language of proteins. tRNAs were those molecules that essentially deliver amino acids to ribosomes as those ribosomes are translating a molecule of messenger RNA to produce a polypeptide chain, that is, to make a protein. Now, tRNAs are able to do this because they have two functional ends, you'll remember. One end of the tRNA molecule is what we called an anticodon. This is a series of bases that will match up through complementary base pairing to the triplet code on the messenger RNA that's being translated. The other end of the tRNA will attach to an amino acid. So the tRNA is a translator kind of molecule. But remember, too, and this is the point I want to get to, that there's a little bit of a problem here that has to be solved in order for this to work. The specificity of the anticodon of a particular tRNA is inherent in the bases of that anticodon because of complementary base pairing between bases of nucleic acids. On the other hand, the site where the tRNA will bind to a particular amino acid is generic. It'll bind any amino acid. So we needed some way to make sure that the right amino acid got connected up to the right tRNA. And by the right amino acid, I mean the one that's appropriately coded for by the codon that tRNA will match up with. This is where an enzyme that has that awkward name of amino acid tRNA synthetase came into play. Let me remind you of how this enzyme works. There's a very large family of different amino acid tRNA synthetases, one for every correct pairing of an amino acid and a tRNA. Each amino acid tRNA synthetase has two specific binding sites. One binding site that recognizes only one kind of amino acid, and another binding site that recognizes the tRNA with the correct anticodon for that amino acid. Thus, the amino acid tRNA synthetase acts as kind of a matchmaker, a molecular matchmaker, making sure that the right tRNA and the right amino acid get put together. Now, in the context of our earlier discussion of information and evolution, this molecular matchup represented an absolutely key step in the creation of orderliness. Orderliness from an otherwise disordered state by imposing the order specified by the base sequence on a stretch of DNA on the addition of amino acids to be added to the growing polypeptide. In other words, we're trying to get the right sequence of amino acids, an ordered sequence. And when we're making a protein, and the only way we could do this is if we got the tRNA to connect with the right amino acid for the codon that it would match up with when it came to the ribosome. So why am I returning to this example now? The reason is simply this. The imposition of this order, which is necessary for the translation of information that's latent in DNA to the functional form of a protein, only occurs with the investment of energy. It takes energy to do this. Now, where does this energy come from? Well, the energy in this case is donated by a molecule called ATP. We've mentioned this molecule before. ATP is short for adenosine triphosphate. ATP acts as kind of a molecular currency that's used to store and transfer energy in all sorts of cellular functions. Now, the reason that we need energy donated by ATP to this process of binding an amino acid to a tRNA is because the bond between the amino acid and the tRNA won't form unless we do add this energy. We need to have a strong bond form here so the tRNA can then take that amino acid to the process of protein synthesis. The bond will not form on its own. All the tRNA synthetase is really doing on its own is matching up the correct amino acid and tRNA. But without the addition of energy, this enzyme can't actually make them hold hands. And that's what the ATP is doing in that particular process. Now, we're going to get back to ATP and how it works in much more detail later. The key point for now is to realize that the energy that's required for this bond is donated by ATP, and by ATP donating this energy, the system becomes just a little bit more orderly. We now have the right arrangement of a tRNA and its appropriate amino acid. This is only a little bit of order, but it's essential for life to occur because it's essential for protein synthesis to occur. The bottom line is that biological systems are very highly ordered things, and this order requires energy to be established. Now, let's take another example, this one from a more recent lecture. This example is from our discussion of how muscles work. And it has to do with the way that the protein myosin, which makes up the thick filaments, you'll recall, of a myofibril, how myosin changes its shape and interacts with the actin proteins that make up the thin filaments of myofibrils. And this interaction between myosin and actin is essential for muscle contraction to occur on a molecular level. Now recall that a myosin molecule has what we call a head region and a tail region, and that the head region can change its position back and forth relative to the tail. I suggested then that you could visualize this by thinking of your forearm as a myosin molecule, with the part of the arm beyond the elbow, from the elbow to the hand, being the head region, and the part of the arm more near to your body being the tail region. Myosin can assume two configurations. The head can either be extended outward or it can be contracted backward. And we, at that time, called this outward extended position of myosin the high energy state of myosin. Now, the way myosin and actin work, remember, is that myosin will extend out in its high energy state, and when it does that, it can attach to an actin molecule on a thin filament. Then it will contract back, it will ratchet back into its low energy state, but because it's attached to the actin, it will pull the thin filament along with it relative to the thick filament, which is what the myosin molecules are made up. And so it's this interaction between actin and myosin that's essential for muscles to be able to change their shape, because it slides past the actin and myosin filaments relative to each other under tension. Now, the point I want to make here is that the way this works depends on the ability of ATP, again, to donate energy to the myosin molecule. The myosin only assumes this extended high configuration state, its head extended outward, once ATP has bound to that molecule and the ATP has essentially cleaved into two separate molecules, with essentially adding one of the phosphate groups, we're going to talk about that in a little while, what that means, to the myosin molecule itself, energizing the myosin molecule and allowing it to extend forward. Now, the way that ATP interacts with myosin, the particular way that ATP is donating its energy to the myosin molecule in muscle contraction, works a little bit differently from the way that ATP specifically is donating its energy to the amino-acyl tRNA synthetase we just discussed. But in both cases, what they hold in common is that the energy that's stored in ATP, and specifically stored in chemical bonds in ATP, is released when one of these ATP chemical bonds, specifically the bond that's holding some of the phosphates that ATP is made up of, is broken. That energy is then released and can be used to change something about another molecule, either creating a bond, as was the case for the tRNA being combined with amino acid, or changing the shape of a molecule, as was the case for the myosin molecule in muscle contraction. In either case, and in the most basic terms, what this added energy is doing is changing the state of the system, and specifically changing it to a higher energy and more orderly state. Now, we could go back through almost any example of a biological process that we've discussed up to this point in the course and make a similar case. Biological systems, as I said, are generally characterized by being highly ordered. On a molecular level, we see this order in terms of the creation of complex molecules from more simple ones, or in terms of functional changes in the shape of molecules once they're built. On the organismal level, we see orderliness in the development of multicellular individuals out of single cells, and in the homeostatic maintenance of this order once it is created. We similarly see orderliness in the structure of populations of organisms, groups of species of organisms in biological communities, and in fact in the structure of entire ecosystems. But, and here's the key point I want to make, all of this orderliness costs something. It costs something to create this orderliness, it costs something to maintain it, it costs something to modify it in a functional way. And in the most basic terms, what all of this costs is energy. We've largely glossed over this issue up to this point in the course, tacitly assuming that all the processes that we've discussed just happen for free. But as we'll see more clearly as we move through this last third of the course, all of these processes, in fact, all of what we would call life itself, depends on having a constant supply of energy in order to create, maintain, and modify a highly ordered structure. If a living organism runs out of energy, it dies. And, of course, when an organism dies, what happens? It loses its orderliness. It falls apart. It decomposes. So, energy is critical for life, and it provides the organizing theme for our next set of lectures. As we've done before, we'll begin our exploration of this theme on the molecular level by considering how energy is stored, how energy is processed, and how energy is produced by cells. First, we have to begin with taking a look at the biochemistry of cells and looking in some detail at the role of energy in enabling this biochemistry to work. Then we'll look at the molecule ATP itself in more detail to gain a better understanding of how this molecule actually contains energy and how it can function as an energy currency in cellular processes. We'll also look in a little more detail at how enzymes work. We've talked about enzymes and their role in biological processes throughout this course, but now we'll provide a capstone to our understanding of enzymes in light of our discussion of cellular energetics. Having established the importance of ATP as a common energy currency in cells, we'll be left with the question of where all this ATP comes from in the first place. Money just doesn't grow on trees, and ATP just doesn't appear magically in cells. This question is going to lead us to a series of lectures on how ATP is produced by cells from the breakdown of other organic molecules, most notably sugar, through processes that we'll call glycolysis, cellular respiration, and oxidative phosphorylation. But even this won't really answer our fundamental question about where all this energy comes from, because we still have to ask where the fuel used to produce ATP comes from. In other words, where do these organic compounds that are broken down when ATP is made of? Or where do these organic compounds come from? The ones that are broken down when ATP is made. This question will lead us to look at the process of photosynthesis, the process by which green plants and a few other organisms can capture energy from the sun, using the energy in sunlight to convert low energy inorganic compounds into high energy organic ones. Once we've established this solid background in cellular energetics, we'll then begin to move up through the hierarchy of biological organization and look at the implication of energy for living systems in general. Now, at this point, what we're going to do is also begin to consider the other half of the theme of this final section of the course, which was what we call namely resources. Living systems need energy to build and maintain order, as I said, but they also need the stuff, the materials out of which they're made. This stuff includes fundamental materials such as carbon, water, nitrogen, phosphorus, as well as a number of other things that are required for life to be put together. As we consider energy and resources at higher levels of biological organization, for example, at the level of populations or communities or ecosystems, we'll see something really interesting. And that is, there's usually not enough energy to go around or enough resources. That is to say, energy and other resources are generally limiting. And the limitation of energy and other resources plays a major role in determining the nature of living systems at all levels of organization. As we look at energy and resources at the organismal level and above in the biological hierarchy, we'll find that we're really talking about the area of biology referred to as ecology. Much of what we study in ecology has to do with how energy and resources are used by different kinds of organisms and how these patterns of use and limitations in energy and resources determine the abundance and distribution of different kinds of organisms. So ecology is where we're going to end this third of the course, ultimately considering the implication of energy and resources for the entire planet, what we might call the biosphere. But we're going to begin this journey next time on the molecular level with some basic biochemistry.